Hi class, this is your instructor, Skylar Huff. We're now here in Chapter 10, and Chapter 10 is about chromosomes, mitosis, and meiosis. Here, pre-existing cells divide to form new cells. This remarkable process enables an organism to grow, repair damaged parts, and reproduce. Cells serve as the essential link between generations. Even the simplest of cells contain a large amount of precisely coded genetic information in the form of deoxyribonucleic acid. When a cell divides, the information contained in the DNA must be faithfully replicated, and the copies then transmitted to each daughter cell. And this happens through a precisely choreographed series of steps. To help you with looking at that, on page 206, please see the figure shown. So here in that figure, that's what you're seeing. DNA is a very long, thin molecule that could easily become tangled and broken, and a eukaryotic cell's nucleus contain a huge amount of DNA. In this chapter, we will consider how eukaryotes accommodate the genetic material by packaging each DNA molecule with proteins to form a structure called a chromosome, each of which contains large amounts of genetic information. So keep in mind, that is genetic information. So here we then consider mitosis, the highly regimented process that ensures a parent cell transmits one copy of every chromosome to each of its daughter cells. In this way, the chromosome number is preserved through successive mitotic divisions. Most somatic cells, or are just regular or ordinary body cells of eukaryotes, divide by mitosis. Mitosis is an active area of biological research, and for good reason. Errors in mitosis can result in a host of disorders and diseases such as cancer, which we'll get to just a bit, and that is a disease condition in which cells divide at an inappropriate rate and become invasive. Thus, a clearer understanding of mitosis has the potential to move our treatment of many diseases. Finally, we will discuss meiosis, a process here, class, that reduces chromosome number by half. Sexual life cycles in eukaryotes require meiosis. Sexual reproduction involves the fusion of two sex cells, or gametes, to form a fertilized egg called a zygote. Meiosis makes it possible for each gamete to contain only half the number of chromosomes in the parent cell, thereby preventing the zygotes from having twice as many chromosomes as the parents. So let us now begin, class, the 11th chapter. This is Eukaryotic Chromosomes. So the major carriers of genetic information in eukaryotes are the chromosomes, which lie within the cell nucleus. Although the word chromosome means colored body, chromosomes are virtually colorless, and the term refers to their ability to be stained with certain dyes. So chromosomes are made of chromatin. That's a material consisting of DNA associated with proteins. So when I get to this, just keep in mind when a cell is not dividing, the chromosomes are present, but in an extended or partially unraveled form. So many times I tell students to think of the chromatin state of DNA as being kind of like a bowl of noodles. So this at the bottom of the screen is what you likely would say, oh yeah, that's chromatin, as composed of what I'm getting to here thereafter. So that chromatin consists of long, thin threads that are somewhat aggregated, giving them a granular appearance when viewed with an electron microscope, as you can see there, class, in figure 10.1, in figure 10.1, I'll continue. And that's figure 411A, not 10.1. So during cell division, the chromatin fibers condense and the chromosomes become visible as distinct structures. So to make sure you can understand things well, this class is at chromatin state. However, that chromatin class condenses into the chromosomal state, which I'm drawing for you now. So of course you have those, the P arm and the Q arm with, of course, they're in the center class, the centromere. The centromere. So this is showing you just what I mentioned. So we have those histone tails being shown there and there, and the entire structure of their class being known as that nucleosome. And you're just seeing how the DNA, the double helix class indicated there, is wound around the cluster of histone molecules. So DNA class, here we are. An organism may have thousands of genes. And just keep in mind, that's a lot of genes, class. For instance, it says here, genes control a cell's activity and are passed on to descendants. So with that in mind, what I'm getting at here, class, is that, for example, humans have more than 20,000 genes that code for proteins. And just keep in mind, class, one gene codes for one protein. So the concept of the gene has changed considerably since the science of genetics began, but our traditional definitions have always centered on the gene as an informational unit. So keep in mind, that is what a gene is. So by providing information needed to carry out one or more specific cell functions, a gene affects some characteristic of the organism. For example, genes govern eye color in humans, wing length in flies, and even seed color in bees. So here, class, getting the prokaryotes and eukaryotes, their cells differ markedly in their DNA content as well as the organization of DNA molecules. For instance, the bacterium Escherichia coli, or what you may call E. coli, contains about 4 times 10 to the 6 base pairs and almost about 1.35 millimeters of DNA in a single, I call it, circular DNA molecule. In fact, class, the total length of its DNA is about 1,000 times as long as the length of its cell. Therefore, the DNA molecule is, with the help of proteins, twisted and folded compactly to fit inside that bacterial cell. However, a typical eukaryotic cell contains much more DNA than a bacterium does. It is organized in the nucleus as multiple chromosomes that vary widely in size and number among different species. Although a human nucleus is about the size of a large bacterial cell, and it contains more than 1,000 times the amount of DNA found in E. coli. The DNA content of the human sperm cell is about 3 times 10 to the 9 base pairs. That's a lot more DNA, you all. Stretched end to end, class, it would measure almost 1 meter long. Remarkably, class, this DNA fits into a nucleus with a diameter only, class, of 10 micrometers, if you can think back to our first lab. So the question is, how does a eukaryotic cell pack its DNA into the chromosome? Well, chromosome packaging is facilitated, class, by certain proteins known as histones. So the histones, class, have a positive charge because they have a high proportion of amino acids with basic side chains. So the positively charged histone associated with DNA, which has a negative charge, and that is all because of its phosphate groups. So those phosphate groups are there to form structures called nucleosomes. So the fundamental unit of each nucleosome consists of a bead-like structure with 146 base pairs of DNA wrapped around a disc-shaped core of 8 histone molecules, 2 each of 4 different histone types. So to help you with what I'm getting at, class, although the nucleosome was originally defined as a bead plus a DNA segment that links to its adjacent bead, Today, class, the term is more commonly referred to the bead itself, meaning those 8 histones and the DNA wrapped around them, as you see it shown here, class. So what I'll get to now, class, would be, of course, chromosome number and the differences in the information content among species. I'm on page 208, the bottom right-hand side. So every individual of a given species has a characteristic number of chromosomes in the nuclei of its somatic cells. However, it is not the number of chromosomes that makes each species unique, but the information the genes specify. Most human somatic cells have exactly 46 chromosomes. But humans are not humans merely because we have 46 chromosomes. Some species, such as the olive tree, for example, also have 46 chromosomes, though some humans have an abnormal number of chromosomes with more or fewer than 46. For instance, it may be class more than, which is 2n minus 1, plus 1, or less than 46 would be 2n minus 1. Each of these class are known as aneuploidies. Each of those are a form of aneuploidy, which of course is off by 1 chromosome. We'll do more of that later. So other species have different chromosome numbers. So a certain species of roundworm has only 2 chromosomes in each cell, whereas some crabs have as many as 200 chromosomes, and some ferns have more than 1,000 chromosomes. So most animal and plant species have between 8 and 50 chromosomes per somatic cell. So quantities above and below these numbers are uncommon. And the number of chromosomes a species has does not indicate the species' complexity or its status within a particular domain or kingdom. Now on to the upcoming section, which is now class section 10.2, found on page 210. It is the cell cycle. So when your cells, class, reach a certain size, they usually either stop growing or divide. And I say this because not all cells divide. Some, such as skeletal muscle and red blood cells, do not normally divide once they are mature. Keep that in mind. Skeletal muscle cells and red blood cells are not going to be typically mitotic cells. Other cells undergo a sequence of activities required for growth and cell division. So the stages through which a cell passes from one cell division to the next are collectively referred as that cell cycle. So the timing of the cell cycle varies widely, but in actively growing plant and animal cells is from about 8 to 20 hours. And the cell cycle consists of 2 main phases, class. They are interphase and Mphase, or mitosis. So this shows you what I just mentioned, class, and just keep in mind that the cell spends most of its time right here in interphase. Most time is spent in interphase. If there were, I guess you would say, a total of 10 hours here, and maybe 1 hour, class, in mitosis or Mphase. So that should help you with that. So most of the cell's life, as I just mentioned, class, is spent in interphase. And to kind of give you, I guess, a bit more information, interphase can be subdivided into, of course, these 3 phases. And before I go too far, let's just go back here. Class, on your lecture tests, you will describe the process of the cell cycle and you should be able to identify those phases. So you will describe those major events of the cell cycle, including the subdividing phases of each of those 2 principal phases being interphase and Mphase. That's your homework for your test. So to be able to describe the cell cycle and mitosis. Getting back to where I left off. So I'll continue here where I left off describing, class, interphase. So here we go. So first things first is G1 or gap 1. So during gap 1, this is that primary growth or first growth. The cell undergoes everyday growth activity. The organelles begin to replicate, and we get our first checkpoint. Next up is S phase or synthesis. Here, class, DNA is replicated. Here, DNA is replicated. Hence the name, class, synthesis phase. Next up is gap 2 or G2. So in gap 2 or G2, what happens to your class is you get secondary growth. So the cell and organelles enlarge, and we get our second checkpoint. So the proteins also begin to synthesize to ensure that everything that is needed class is here in preparation for that cell to divide. So this class shows you what interphase looks like. You have, of course, that plasma membrane, the cytoplasm class, and you just have everything that there is needed. And yes, the chromatin state of DNA is still there. And it also shows you all centrioles being located there. More about those here shortly. Now, class, getting on to M phase or mitosis. So what's happening here, class, is that, well, M phase involves two main processes. We call it cytokinesis. So that is, class, the division of the cytoplasm. And I'll mention also karyokinesis, which you may say, what? Yes, karyokinesis. And I mentioned karyokinesis, and I'll write it out to make sure you know what I'm saying. That would be, class, the division of the nucleus. So cytokinesis and karyokinesis. Keeping in mind, class, that mitosis or M phase is the shorter of the two phases. So with that, there are, it says 5 stages here in your text. I'm going to distill down those 5 stages, class, to what I call 4 stages. So I'll call those prophase, metaphase, anaphase, and telophase, or telophase, however you like to say it. So to keep you where you should be, class, at the end of M phase, at the end of mitosis, what will be created, class, is, of course, 2 cells. And those 2 cells, class, are genetically identical. So as it states, this is that nuclear division, class, that produces 2 nuclei containing chromosomes identical to the parental nucleus. And in case you're wondering, okay, yes, Huff, you're going through the cell cycle. Why is this important? Well, the cell cycle, if you don't know yet, class, it is important because it's that integral part of normal growth of our cells, normal growth of the size of your organs, and even repair of existing structures. So if you were to have a cut or a scrape, and it began to bleed, and even if it did not bleed, class, a cut or a scrape has to be repaired, and this is a way in which it's repaired by the way of the cell cycle, your cells dividing normally. And the same can be stated, class, by, I'll just go right to it, class. It says normal growth, such as size of organs. So as we, class, get bigger and bigger and bigger, whether it's getting fat, getting tall, getting small, I'm kidding, but class, it's because of the division of these cells. Let's now continue. So I'm here, class, in prophase, and I'll say again, on your lecture test, you'll describe, class, the cell cycle in its entirety. So that includes, class, both phases and those subdividing phases. So from interphase, it includes gap 1 or G1, S phase, and G2. And here we're now in mitosis. That does include, class, prophase, metaphase, anaphase, and telophase. I'm here, class, in prophase. Let us begin. In prophase, class, the nuclear envelope disappears. Spindle fibers form from that disappearing nuclear envelope, class, by one of those microtubules. And what then happens, class, is the centrioles move toward the poles. So as that happens, the chromatin condenses, class, into the chromosomal state, and they're now called sister chromatids. And I got to make sure I stress, class, it's duplicated chromosomes. I repeat, it's duplicated chromosomes. So if I were to draw this that looks like that, draw this that looks like that, we now, class, have duplicated chromosome number 1, so to speak. And then the rest of which, class, that I had not mentioned would be that just attached to each centromere, which I'm going to change my color here to make sure you can see. The centromere, class, is there of that duplicated chromosome. So attached to the centromere, class, is that kinetochore to which those microtubules that I mentioned about spindle fibers, class, can bind. So to say it again, class, it's that centromere that holds together those sister chromatids. And I guess I should draw this, and maybe I'll draw it in another color. So this, class, is one of the 2 sister chromatids, and this, class, is the other sister chromatid, and I'll just draw, class, here in a blue, the centromere, that, of course, is holding the sister chromatids together by way of cohesions. This class is prophase. So with that, animal cells class have a pair of centrioles in the middle, and this class is what will hold those spindle fibers. And here shortly we'll get to holding the chromosomes too, by way of, well, in part of the cell. So this class is it. This is what prophase looks like. So if you look closely, I mentioned a second ago, we have the chromosome here, chromosome 1. Well, this is also chromosome 1. So now that we have those two, let's go back to where our test was here to show you yet again. So you have those pairs of duplicated chromosomes by way of the sister chromatids. So let's continue on now, class, because it's the sister chromosomes that make up the duplicated chromatid, or duplicated chromosome. So I guess I'll say this is a duplicated chromosome. So that's a duplicated chromosome as well as that one in red on the left. Let us now continue. So look for this on your test. The figure should look very, very similar, class. So here with Prometaphase, class, this, it states, begins when the nuclear envelope breaks down. I just mentioned that, class, moments ago. So just keep in mind that I said I'm just going to give you those four phases: prophase, metaphase, anaphase, and telophase. So let's continue on now, class. Here is metaphase. So in metaphase of mitosis, what happens, class, is that the chromosomes line up at the metaphase plate, spindle fibers attach to the centromere and the centrioles, and this happens, class, by way of that kinetochore signal. And lastly, I'll say, this is where we have our third, class, our third, and I'll call it the third and final checkpoint. The third and final checkpoint is here, class, and I'll get back to that momentarily. So with this being stated, class, the cell will arrest itself here to ensure that it has everything it needs before it proceeds, class, to the next phase. And I say this now because if the cell were to continue and not have what it needed, it would divide, class, no matter what. So this class is metaphase, and as you see it, class, you have 1, 2, 3, 4 chromosomes lined up here, class, at the metaphase plate, as I mentioned earlier. And it's just that imaginary midline. The imaginary midline. So again, I said centrioles have moved toward the poles, as you all see them. And I say spindle fibers, by way of the microtubules, class, attach from centriole. I'll grab a color here. From centriole to centromere to centriole. And that's, of course, for each of those 4 chromosomes. And one thing that I should go backwards and do, class, and let you know that this happens in our somatic cells. And our somatic cells are diploid cells. So diploid, I'll write it out, Diploid, diploid cells. Diploid cells have 2 complete sets of chromosomes. So the diploid number for this cell is 1, 2, 3, 4. Since there are 4 chromosomes, class, in this case, the diploid number is 2n equals 4. Keep that in mind because I'll use this class when I get to the process of meiosis here in a moment. So this class shows the very same. You have the sister chromatids, class, those chromosomes, those duplicated chromosomes, the metaphase plate, which I mentioned, class, at that cell's midplane. And I've said by way of that kinetochore, the spindle fibers, class, can attach themselves from centriole to centromere to centriole. That's the process, class, and that's how it looks. Look for it in your test. Up next is anaphase. I'll be brief. The sister chromatids move toward opposite poles of the cell, and the cell elongates. Class, it's preparing for division. So as this has happened, class, each of those sister chromatids are now actually chromosomes. So I'm just going to draw a line here for teaching purposes, class. Each end of the cell, class, now has enough information to be its very own cell. Exactly the same, class, as a cell that we began with. This is anaphase of mitosis. Here, class, is telophase. Telophase, however you like to say it. I'll put it this way. You can learn this if you've learned prophase by just stating that telophase is the opposite of prophase. Now, please be blunt and honest. Class, do not write, oh, it's just the opposite. What I'm getting at is once you've learned prophase, you can say that, okay, I know telophase because it's the opposite class of prophase. I'll begin. So the nuclear envelope reappears, and just keep in mind it's built by the broken down spindle fibers. So chromosomes here, they decondense because, hey, they got to get back, class, to that chromatin state of DNA. So what I'll get to now, class, is a couple of differences. So the cleavage furrow forms, class, and just keep in mind that it is a cleavage furrow in animal cells. Animal cells, class, have that cleavage furrow you see here. However, if this were to be plant cells, class, and plant cells is called a cell plate. So the cell plate forms in plant cells. For instance, let's just say this cell here is dividing, and of course this cell here is about to divide. The cell plate is right there, and that of course is what will be occurring. So please do not forget that. So of course we have both karyokinesis, class, the division of the nucleus, and cytokinesis, the division of the cytoplasm, and what we're left with, class, are two genetically identical cells. So this is, class, the cell cycle. So there we have it. On the test, you'll describe it. So now, class, being shown is what I just mentioned, class, moments ago. So these are our plant cells. Notice there are cell walls there. So of course, with that being the case, the cell plate is now forming as it states, and yet in still, class, we get two cells. So there it is, the cell plate yet again shown. This is the process. So just keep in mind, class, each daughter's nucleus receives exactly the same, I repeat, exactly the same number and kinds of chromosomes of the parent cell. And this is because, class, this is nothing more than a replica. I said normal everyday growth and repair. So the organelles are a portion, class, that the daughter cells received, and the mitochondria and chloroplasts, they do, class, contain their own DNA, which of course formed by the division of the previously existing mitochondria or plastids in the case of the chloroplasts. So let's get to prokaryotes here. We'll be here briefly. So prokaryotes, though they lack nuclei, they still divide by way of the process known as binary fission. Prokaryotes do. So bacteria and archaea contain much less DNA than do most eukaryotic cells, but precise distribution of genetic material into two daughter cells is still a formal process. So the prokaryotic DNA usually consists of that single circular chromosome that is packaged and associated with proteins too. So although the distribution of genetic material dividing the prokaryotic cell is a simpler process than mitosis, it is nevertheless, class, very precise to ensure that the daughter cells are genetically identical to the parent cell. So prokaryotes reproduce, class, asexually by way of binary fission. And it's this process in which one cell divides into two identical daughter cells, two identical bacterial cells. So the circular DNA molecule, it replicates, resulting into two identical chromosomes, and DNA replication begins at one site on the bacterial chromosome, called the origin of replication, and the DNA synthesis proceeds from that point, class, in both directions until they eventually meet. So just keep in mind, class, it's the very same process, just a bit simpler, going from one cell, class, to two cells, each exactly like the first cell. So notice here, class, there are about to be now two copies of those circular chromosomes, the double helices, here in the bacterial cell. So with regulation of the cell cycle, I'll be here briefly, class. It happens by way of cyclins. And it states that under optimal conditions, the length of the eukaryotic cell cycle is constant for a given cell type. In your cells, class, they should divide an approximate 100 times. Of course, as we do age, I did say as we age, class, they'll divide lesser and lesser than, of course, the 100 times. But since we are pretty young, I said the cells divide an approximate 100 times. And then, of course, they die. They go through the process, class, of apoptosis, which is nothing more, class, than programmed cell death. Because, hey, all cells must die at some point because they don't live forever. So the cell cycle checkpoints I mentioned, class, they ensure the events of that particular stage have been completed before that next stage begins. I mentioned this, class, throughout the process of mitosis. So if a gene encodes for a molecule involved and the checkpoint is defective, cancer or a serious disease can result. And let's get to those things now. So what happens is we have these things called CDKs. They're called cyclin-dependent kinases. So to get you to where I'm going, class, is I'll say that cyclins are proteins. And those are the proteins that control regulation of the cell cycle. So cyclin production begins in S phase, and the cyclin production, class, increases in concentration until anaphase. So what then happens, since they're called cyclin-dependent kinases, is that the cyclins bind with CDK, the cyclin-dependent kinases, and as that happens, they eventually, class, form to get to what is known as maturation promoting factor, and it's MPF. And once maturation promoting factor reaches threshold, we get, of course, mitosis. So at the end of metaphase, I mentioned, what then happens is we get anaphase promoting complex, which initiates anaphase. So we must have anaphase promoting complex to initiate anaphase. And it's all based on, of course, that maturating promoting factor by way of those cyclin-dependent kinases. So given this, yes, drugs can be used to, of course, arrest cells at this point or that point of the cell cycle. But I won't get too much of that, not at this moment. So next we're here, class, with sexual reproduction and meiosis. Now we're at sexual reproduction and meiosis. So with asexual reproduction, it's a single parental cell that splits and produces that clone. It's rapid and efficient. However, class, what happens is that organisms here going through asexual reproduction, they're likely, class, going to be in an environment that is favorable to them. And I say that because they're not changing over time, i.e. they're not evolving, they're not adapting. And it's because they're clones. They're all the exact same. Watch out, bananas. I did say that. The bananas we're eating, class, are clones. They're all the same. So please don't take anything, class, into any banana fields or orchards. We might have no more bananas ever. Thanks a lot, global travel. But of course, with sexual reproduction, class, in which you have that union of the egg and sperm, that from the zygote, this, class, results in genetic variability, giving us genetic variation in an offspring. This, class, is what does indeed favor adaptation. This, class, is what indeed that allows change. It allows, class, species to evolve. And of course, evolution is all about change over time. Let us now continue. So although the cells of reproductive processes, they vary greatly among different types of eukaryotes, there are just two basic types of reproduction, which I just gave you, class. They're known as asexual reproduction and sexual reproduction. So let's get into the process, class. I began here on section 10.4 on page 219, and now, class, I am continuing. So I'm here, class, and I'll just state that usually two parents contribute to the gametes, and in some cases, in animals and plants, the egg and sperm cells are the gametes and are fertilized eggs and the zygote. So I'll next get to homologous chromosomes. So homologous chromosomes, class, is just a pair of chromosomes, and they're similar in size. So, as I was stating, homologous chromosomes are a pair of chromosomes, and they're similar in size, shape, and position. And as I've mentioned, they're similar in the size, shape, and position of the centromere. So as I begin to draw here, of course, we have that, and we have this. And then I'll draw in the center, the centromere. So I say, similar in size, shape, and position of the centromere. So class, we have 46 chromosomes. And those 46 chromosomes, class, are made up of 23 homologous pairs. So that just means, of course, we have chromosome 1. And, of course, we could continue on and say there is also chromosome 2, and chromosome 3, chromosome 4, and so on and so forth. So there are 23 homologous pairs and 46 chromosomes. So just keep in mind, class, the terms I mentioned last lecture, just before now. So diploid chromosome number means that that cell contains 2 complete sets of chromosomes, whereas the haploid number, which this is new, I did not mention class haploid before. So the haploid number, class, states and means that that cell or nucleus has a single set of chromosomes. So us being Homo sapiens, us being, of course, as we are, class, our diploid chromosome number, class, is 46. Our diploid number is 46. And, of course, our haploid number is 23, which, of course, is half of 46. So gametes, class, are haploid. Keep that in mind. The gametes are haploid, i.e. the sperm and egg. They're haploid. So by way of fertilization, class, each gamete contributes N plus N, giving us 2N. And that would be, of course, that zygote. That 2N, class, would, of course, represent that zygote from the fertilization event, from that sperm cell, and, of course, this egg cell. So the zygote would then, class, divide by mitosis, or cleavage, going through cleavage. So all somatic cells receive the same diploid set of chromosomes. Let us keep going. I won't mention much about polyploidy because it does not happen in animals, but of course, that's when you have multiple sets of chromosomes, such as 16N, class, seen in some plants, or even class 8N. And I like to think of Fragaria, the strawberry, or even class a lot of fern species, that have, of course, polyploidy. The process. So meiosis produces haploid cells with unique gene combinations. So it is a cell division that reduces chromosome number by 1/2. So keep this in mind, class, that what meiosis does is it's comprised of 2 divisions, and you take what is a diploid cell, so I just draw that cell and draw 2N, and what happens with that diploid cell is that it then, class, will divide to yield, class, 4 haploid cells. And this, class, is really and truly the process as it occurs, as I'm drawing it for you all now. So we begin with that 1 diploid cell, and we end up, class, with 4 haploid cells. So to be more specific here, class, I say that meiosis occurs, and it undergoes 2 nuclear and cytoplasmic divisions. So by way of meiosis 1 and meiosis 2. And each of which, class, include prophase, metaphase, anaphase, and telophase. And I probably shouldn't do this right now, but it's because I want to make sure you get the processes. So here, class, is meiosis. It's spelled incorrectly. I said that doesn't look right. Try that again, Huff. So I say again, here is meiosis. Yeah, now that's better. And on the right-hand side, I want to draw, class, mitosis. Mitosis looks just like this for learning purposes. 2N, 2N. So I hope you can now see that mitosis is what, of course, makes exact replicas, duplicates things, as opposed to meiosis, which halves chromosome number. Let's continue now. So we have those 2 nuclear and cytoplasmic divisions, class. So what happens by way of meiosis 1 is homologous chromosomes. They pair, they separate, and move into different nuclei. By way of meiosis 2, we have sister chromatids that make up each duplicated chromosome that separate from each other and are distributed, class, into two different nuclei. So let's get to the process now to make this make a lot more sense. And you're seeing now pretty much what I got to not long ago. So we have interphase, which occurs here, and yes, the cell must still, class, with the process of replication. And no, there will never be 92 chromosomes. There will never be 92 chromosomes. However, there will, class, be 23 tetrads, which we haven't gotten to yet, the term tetrad. So there will be 23 tetrads, or you can very well just call it, class, there will be 92 chromatids. I said chromatids. I will go with that, but never, class, 92 chromosomes. So let's get to the process now. So you see Meiosis I, we have those homologous chromosomes that then separate, and then Meiosis II, in which those sister chromatids separate. Continuing now. So Prophase I, class, what happens here is a two-piece. They're called synapsis and crossing over, class. This is where it gets to be one of the most amazing things on earth. So here, synapsis, class, happens when you take a pair of homologous chromosomes. And of course, each from one parent. So we now have, class, what is known as a tetrad. I say again, we have a tetrad. So I'm going to draw these a little differently than I've drawn them before. So we have this, that's here. And then, of course, we have that's there. And that, class, is what I'm referring to as being a tetrad. So maybe this is chromosome 3. Maybe they're both from chromosome 3. And that's our tetrad. And I'll just write it in the center. So I'll continue now, class. So we have, class, crossing over that occurs. And the reason I say crossing over is because it is that exchange of genetic material. I say again, it's the exchange of genetic material producing new combinations of genes. And it happens, class, at the chiasmata. It happens at the chiasmata. So I'm going to draw this very same thing I have drawn, class, here before. But what I'll do now is I'm going to draw them as they come together for the process of synapsis. So there is that, and there is that. So this is that very same thing, and I want to draw using, I guess, my orange to add my centromeres here. So there are my centromeres there, and of course, there they are there. So it's, class, where they overlap, meaning synapsis has occurred. So where this tetrad overlaps is known as the chiasma. So there is a chiasma here, and there is a chiasma there. So at those chiasmata is where, class, you get that exchange of genetic information. Meaning those new gene combinations, class, are why I am 5'7". They are why you are, I don't know, 5'2". They are why you are 6'6". They are what make us different. This is why you may have blonde hair, you may have brown hair, you may have red hair. You may have blue eyes. I have brown eyes. You may have hazel eyes, meaning everything that exists that is different, class, in part because of this. And this is just one of three ways, class. I'll now continue. So as you're seeing it, this is the same thing, class, I drew for you moments ago, giving you the homologous chromosomes that, of course, pair up to create that tetrad and then allow them to synapse, to come together, and allow the process to occur called crossover or crossing over, to exchange, class, and create those unique gene combinations. So I'll now continue, class, meaning you're here, class, from prophase, we're now going to metaphase. So, and it's not just metaphase, but it's metaphase 1. So the tetrads align at the metaphase plate, as of course you saw before. So what happens now, class, is known as independent assortment. So it's the way in which those chromosomes assort themselves at the midplane, and it happens, class, independently of one another. Class, that's the second thing I repeat, that's that second part that provides us with variation, with differences. And then thirdly, class, I get down to Anaphase 1. So here in Anaphase 1, what then happens is that paired homologous chromosomes separate and move toward the poles. Class, this is called segregation. So as they begin to segregate themselves, class, that happens independently of one another as well. Meaning that's that third way, class, in which meiosis provides us with these differences, the variation we get. So it's now that each pole has a random combination of maternal and paternal chromosomes. And I'll be honest, class, it's right now that we have had that reduction division. So what I'm getting at, class, is the cell is not any longer, what I call diploid, class? The cell is actually now haploid. It is now in because of that reduction division. So we'll next get to what is known as telophase 1. So each cell contains, here we go, a haploid number. Each cell contains a haploid number of chromosomes. And this, yes, is a haploid number of duplicated chromosomes, but these chromosomes, class, are now different. And if you don't know what I mean, look, there's a little purple tip here, there's a little blue tip there, meaning that's just to show you. But they are different, class, because, of course, the crossing over that occurred by way of synapsis. So we have 23 duplicated chromosomes. There are 23 duplicated chromosomes, or you could say, class, that's chromosomes, or you could say, class, that there are 46 sister chromatids. So there you have it. There we go. So Meiosis II. To help you out, class, with your learning, you could definitely say that Meiosis II is very similar, class, to the process of mitosis by way of prophase, metaphase, anaphase, and telophase. For instance, there's not much, class, happening, of course, in prophase 2, but I will say, class, there is no pairing of chromosomes here in prophase 2, and there is no crossing over. And do keep in mind, class, this is prophase 2, and yes, there are two cells, meaning it already divided once, so there are two cells now. So in metaphase 2, there is a bit of a difference, class, in that the chromosomes, they independently assort themselves, class, at the metaphase plate. So hey, gaining variation yet again. And then we get back to anaphase 2, in which those sister chromatids separate and move to opposite poles. Well, they're now, of course, referred to as being chromosomes. They're now actually chromosomes again, you all, and not sister chromatids. But yet again, they independently of each other segregate themselves. So that is, again, class, providing more variation. From here, class, we get to telophase 2. And we have, class, one chromosome from each homologous pair here. And what I'm stating is that we now, class, have what I refer to as being 4 cells with 4 unique haploid nuclei. So each 4 of these cells are different from each other and different, class, from that cell we began with. So in the end, class, we will then have what are known as 4 gametes. I would say especially, class, especially in the case of males. However, class, females would only have, class, a single egg. Females would only have a single egg, and we'll do more with that, class, here thereafter. And in the event, class, this were to be in the plant kingdom, these would be spores. So again, they are 4, I repeat, there are 4 genetically different haploid cells. And I'll look for that on your test when you describe the process. So to end the process, just keep in mind there are 2 divisions of meiosis that yield 4 haploid nuclei with each different combination of genes. Each of those are different. They are not at all the same. And in fact, I sometimes tell the students, grab a calculator, type in 2 to the 23rd power. Please. Type in 2 to the 23rd power. So by typing in 2 to the 23rd power, And I'm waiting for your answer, actually. If you have no calculator, class, here's your answer. That's just how many class gene combinations are possible by way of meiosis. In the meantime, class, this is why we're different. This is why everyone here on Earth are different. So with this, mitosis class has a single nuclear division which produces two daughter cells that are genetically identical to each other, and there is no cell, as opposed to in meiosis, we have a diploid cell that undergoes genetic rearrangement by way of course, synapsis, and then crossing over by way of independent assortment class, and of course, segregation that occurs. We get those two successive nuclear divisions class, resulting in the formation of four genetically different haploid daughter cells. So with sexual life cycles class, and I'll end this lecture here shortly, I'll just state class that gamete formation class is known as gametogenesis, the formation of gametes. And I hope you can follow me here when I say spermatogenesis, which of course is the formation of those four haploid sperm cells, or of course each cell that enters into meiosis. So yes, we have what is known as a primary spermatocyte that goes through the process class of creating the gametes, known as, of course, spermatogenesis. Well, with oogenesis class, meaning this is how we get our germline cells, well, to create the ova. So it's one egg cell for every cell that goes through meiosis. For instance, I'll be brief here. Let's just draw it this way. So that class, the primary oocyte, we get this division here. And then we get that division there, that division there, boom. So those three that are going to be to that left-hand side class, these are what I'm referring to as being those polar bodies. And that class will be that one egg. But then don't get too bogged down here, class, meaning this is what happens by way of oogenesis, the primary oocyte. And I'm saying don't get too bogged down here, class, because, in fact, this doesn't go that far, unless, of course, fertilization happens anyway. In the meantime, class, this is the process of meiosis. And don't forget that we also have what is known as the alternation of generations, but I'll do more of that class a bit later. So please study well. This has been Chapter 10. I am your instructor, Scholar Huff, and if you need help with this, reach out to me. Call me on my office telephone number, stop by the office, or even email me. Thank you all for listening, class.